Following our initial discussion on seed viability, we will now turn our attention to the methods that have been developed to test it. There are several approaches available, but in this lecture, we will focus on the two most widely accepted, the standard germination test and the tetrazolium test. These methods form the foundation of seed quality assessment and will give us a clear understanding of how viability is measured in practice. Let's begin with the germination test. According to the Association of Official Seed Analysts, several standards must be followed when conducting germination tests. The first is separate testing. Seeds of agricultural crops, vegetables and herbs, flowers, and trees or shrubs should be tested separately. The second is accurate identification. Both the scientific and common names of the species must be provided. The third rule concerns substrate specification. The substrate used for testing seed viability should always be indicated. Finally, there are substrate requirements. Substrates must be non-toxic to germinating seeds, free of molds, microorganisms, and their spores, and able to provide adequate aeration and moisture for germination. Why does substrate choice matter? The substrate is the immediate environment in which seeds attempt to germinate. If it contains toxins, pathogens, or lacks proper aeration and moisture, seeds may fail to germinate even though they are viable. This can lead to misleading results, underestimating the true quality of a seedlot. By standardizing substrate conditions, analysts ensure that differences in germination reflect the seed's inherent viability and not flaws in the testing environment. This table summarizes the germination testing requirements for 7 different species. It highlights how testing conditions are standardized, yet also tailored to the biology of each crop. For each sample, we list the species and its common name, the substrate used for germination, the temperature requirement, the timing of the first and final counts, and any additional specific requirements for each species. Notice how the choice of substrate varies among species. Some germinate best on petri dishes lined with moist paper towel, abbreviated as P, others on butter paper, and still others on paper towels. Temperature is another critical factor. Each species has an optimal range, and deviating from it can significantly reduce germination percentages. The first and final counts serve as standardized checkpoints. The first count records early germinators, while the final count ensures that late germinating but still viable seeds are included. Finally, specific requirements such as light exposure or pretreatments demonstrate how protocols are adjusted to accommodate the unique characteristics of different seeds. Altogether, this table illustrates why AOSA rules are so important. They ensure that no matter where the test is conducted, results remain consistent, comparable, and biologically meaningful. Aside from the germination test, another important method for estimating seed viability is the tetrazolium test. This technique was developed in the early 1940s by Professor George Lacan at Hohenheim University in Germany. Unlike the germination test, which requires seeds to grow into seedlings, the tetrazolium test works by distinguishing between living and dead tissues of the embryo based on their respiration activity in the hydrated state. The principle behind the test is straightforward. It relies on the activity of dehydrogenase enzymes, which serve as indicators of respiration and therefore of seed viability. When seeds are soaked in tetrazolium salt solution, these enzymes react with their substrates and release hydrogen ions. The hydrogen ions then reduce the colorless tetrazolium solution, transforming it into a red compound called formazan. As a result, living tissues of the embryo stain red, while dead tissues remain unstained. This clear visual contrast allows analysts to quickly determine whether a seed is viable without waiting for it to germinate. In this way, the tetrazolium test provides a rapid and reliable assessment of seed quality, making it a valuable complement to the standardized germination test. The general protocol for the tetrazolium test begins with imbibition, where seeds are placed on a moist substratum to allow complete hydration of all tissues. This step is essential because the tetrazolium solution can only penetrate and react effectively when the seed tissues are fully hydrated. In some cases, seeds must be cut or punctured to ensure that the tetrazolium salt can reach the embryo and all internal tissues. Once prepared, the seeds are immersed in a tetrazolium solution and incubated in an oven at about 35 degrees Celsius. During this period, the solution interacts with the dehydrogenase enzymes in living tissues, leading to the development of a red coloration. This staining process, known as formazan formation, provides a clear visual distinction between viable tissues, which turn red, and non-viable tissues, which remain unstained. When interpreting tetrazolium test results, it's important to remember that not all tissues stain in the same way. Sound, healthy embryo tissue tends to absorb the tetrazolium solution more slowly, and as a result, it often develops a light red color. In contrast, tissues that are bruised, aged, frozen, or otherwise damaged usually stain more intensely because of their altered respiration activity. Equally important is the pattern of staining. It's not just about whether the tissue turns red, but where and how the staining appears. Correct interpretation requires a solid understanding of the different parts of the seed and their functions, since the viability of the seed depends on whether the essential structures of the embryo are alive and functional. For example, if we look at the pictures of seeds on this slide, you can see the embryo axis and the cotyledon, both of which must be evaluated carefully. Finally, it's worth noting that many seeds are not simply completely alive or completely dead. Instead, they may show partial viability, with some tissues staining red while others remaining unstained. This makes interpretation a nuanced process that requires both technical skill and biological insight. When we interpret tetrazolium test results, the location and extent of staining are what determine whether a seed is considered viable. Seeds that are completely stained are classified as germinable, since all essential tissues are alive and functional. A good example of this seed is a seed labeled number 1. In some cases, we see only minor unstained areas. For example, seeds with small unstained spots on the cotyledons or even at the extreme tip of the radicle are still considered germinable because these regions are not critical for seedling development. Seeds labeled 2 to 6 are good examples of this category. However, when unstained areas occur in essential structures, such as the radical hypocotyl axis or the juncture where the cotyledons attach, those seeds are classified as non-germinable. Damage in these regions prevent the embryo from producing a normal seedling. For instance, if the unstained area bisects the radical hypocotyl axis or affects the plumule, the seed cannot develop properly and is therefore non-viable. Seeds labeled 7 to 15 illustrate this. This progression moves from the fully stained and germinable seeds to partially stained but still viable ones, and finally to critically unstained regions that render seeds non-germinable. It highlights the importance of both staining intensity and anatomical context. It's not enough to simply note that red coloration is present. We must also evaluate where the staining occurs in order to make an accurate judgment of seed viability. This slide provides additional examples of tetrazolium staining results in Arabidopsis, barley, and pepper seeds. In the left panel, you can see that the viable seeds are completely stained red, while the dead seeds show no staining at all. The next image illustrates variable staining, where we observe a mixture of abnormal, viable, and non-viable seeds. In the barley panel, most seeds display an evenly stained embryo axis, which indicates viability, except for these two examples shown at the bottom where staining is incomplete. Finally, we have an image of differentially stained pepper seeds. Here, the embryo is intact as indicated by the white arrows, but the partial staining indicates that the seed is no longer viable. One of the main advantages of the tetrazolium test is its rapidity. Unlike germination test, which may take several days or even weeks, the tetrazolium test can provide results within a matter of hours. This makes it especially valuable when quick decisions about seedlots are needed. Another important advantage is its usefulness in assessing dormant seeds. Because the test measures tissue viability directly, it can reveal whether dormant seeds are still alive, something that a standard germination test cannot always show. The primary disadvantage, however, is that the test requires considerable experience to interpret correctly. The staining patterns can be subtle, and distinguishing between viable, partially viable, and non-viable tissues demands both technical skill and a solid understanding of seed anatomy.